Hey everyone, and welcome. Today, we are diving deep, really deep, down to the microscopic level to figure out one of the most amazing processes in all of biology, how tiny little microbes power their world.
You know, it's a wild thought, isn't it? How does that sugary treat actually turn into the energy you need to keep going? And if we can do that, how does a single-celled organism with no stomach or lungs manage to fuel its entire existence? Well, it all comes down to a process called metabolism. So here's our game plan. First, we'll look at the cell's energy economy. Then, we'll meet the tiny little workers that make everything happen, the enzymes. After that, we'll follow a single sugar molecule as it gets split for the first time. Then comes the big finale, the powerhouse payoff. And finally, we'll see what happens when life has to find a way to thrive without oxygen.
All right, first stop, a cell's energy budget. The best way to think about a cell is like a bustling microscopic city. It's always busy, constantly tearing things down for raw materials and energy, and then using those to build new structures. It's a non-stop cycle of income and expenses. And this whole citywide operation is called metabolism. It's split into two sides of the same coin. You've got catabolism, that's the demolition crew, breaking down complex things like sugars to release energy. And then you have anabolism, that's the construction crew, using that energy to build everything the cell needs to live and grow.
So what's the cash that this city runs on? Well, it's a fascinating little molecule called ATP, adenosine triphosphate. You can think of it as the universal dollar, or euro, or yen of the biological world. Every time a cell breaks down food, it's basically stuffing its wallet with ATP to pay for, well, everything else.
An even better way to picture it might be as a tiny rechargeable battery. When the cell needs to do some work, it plugs in an ATP and uses its charge, which turns it into a dead battery called ADP. Then, the energy from our food comes along and recharges that ADP right back into a fully powered ATP. It's this frantic, endless cycle happening millions of times a second.
Now, you might be wondering, how do all these chemical reactions happen so fast and so efficiently? Things don't just break down and build themselves. They need a little help. So let's meet the real heroes of our story, enzymes.
Think of enzymes as the most dedicated, hyper-efficient workers you've ever seen. They're biological catalysts. They grab the raw materials, which we call substrates, and they perfectly position them to react, turning them into products way, way faster than would ever happen naturally. And the best part? They clock out completely unchanged, ready for the next shift.
Here's how it goes down. Step 1. The substrate docks into a perfectly shaped little pocket on the enzyme called the active site. Step 2. They form a temporary little team, the enzyme-substrate complex. Step 3. The magic happens. The substrate is converted. And finally, Step 4. The newly made products are released, leaving the enzyme ready for the next customer.
Okay, so if these enzymes are so good at their jobs, wouldn't things get out of control? A cell needs to manage its production, right? It can't just have all its factories running at full tilt 24/7. And that's where inhibitors come in. They're the managers that tell the workers when to take a break.
And there are two main ways to hit the bricks. On the left, you have what's called a competitive inhibitor. It basically looks just like the real substrate and plays a game of musical chairs, stealing the spot in the active site so the real substrate can't get in. Then you have the non-competitive inhibitor. This one is a little sneakier. It binds to a totally different part of the enzyme, and by doing so, changes the shape of the active site so it just doesn't work anymore.
All right, so we've met the currency, we've met the workers and the managers. Now, let's actually follow our hero, a single molecule of glucose, or sugar, on its journey to becoming energy. And that adventure starts with a process called glycolysis.
Glycolysis literally just means sugar splitting, and that's a perfect name for it. This all happens in the main body of the cell, the cytoplasm. And here's the cool part. It doesn't need any oxygen at all. Now, you know the old saying, you have to spend money to make money? That's exactly what the cell does here. It invests 2 ATP molecules to get the process started before it gets to the payoff phase.
So after all that splitting and rearranging, what's the net profit? Just 2 ATP. Yeah, I know it doesn't sound like a lot, but here's the thing. It's incredibly fast energy. Think of it like the cell's pocket change. It's not the big prize, but it's the crucial first step to unlocking a treasure chest of energy later on. So, we just turned our one glucose molecule into 2 smaller molecules called
pyruvate. And these little guys are packed with potential. Now, it's time to take them to the cell's central power plant to really cash in.
But before we get to the main event, there's a quick pit stop. Each of those pyruvate molecules gets a little haircut, turning into a 2-carbon molecule called acetyl-CoA. So, what happens to that carbon that got snipped off? It's released as CO2. And get this, this is the exact same carbon dioxide that you're breathing out right this second. Pretty amazing, right?
That acetyl-CoA then jumps into something called the Krebs cycle, which you can think of as a molecular furnace. It just completely dismantles what's left of our sugar, releasing even more CO2. Now, it only makes another 2 ATP directly, but its real job, its most important job, is to load up a ton of energy onto special carrier molecules. Think of these like tiny rechargeable power banks, getting all charged up for the final step.
And here we go. This is the grand finale, the main event. It's called the electron transport chain, and this is where all those little power banks we just charged are going to go to finally cash in their energy.
And just look at this chart. This really tells the whole story. Glycolysis gave us a measly 2 ATP. The Krebs cycle gave us another 2. But the electron transport chain, it turns out a mind-blowing 34 ATP. I mean, it's not even close. This is the powerhouse. This is why complex oxygen-breathing life is even possible.
Okay, so that's awesome if you have oxygen, but a huge number of microbes on our planet live where there's no oxygen at all, and even our own muscle cells run out of it when we're sprinting. So what's plan B? How do you make a living when you can't breathe?
This table breaks it all down beautifully. It all depends on what the cell uses to, well, take out the trash at the end of the line, what's called the final electron acceptor. Aerobic respiration, our method, uses oxygen and gets that huge 38 ATP payoff. Anaerobic respiration uses other things like nitrate or sulfate and gets a decent but smaller return. And then you've got fermentation, which gets a tiny yield of just 2 ATP.
Now this is a super important point, and one that often gets misunderstood. The whole point of fermentation is not to make more ATP. It actually makes 0. Its only job is to recycle those energy carrier molecules, the power banks, so they can go back to glycolysis and keep that first little process running. It's a survival hack that lets cells keep making that tiny profit of 2 ATP, and it's what yeast uses to make our beer and bread.
So when you really step back and think about it, a single tiny microbe is running an incredibly complex and adaptable power plant inside its tiny body. It's just staggering, and it really makes you wonder, if they've mastered something this fundamental to life, what other amazing things are they doing all around us, right under our noses?